Today I want to show you some results from my PhD project, which is mainly focused on chromatin organization analysis. So this is the roadmap of my presentation. You can follow the roadmap in the upper right side of the slides, and it is divided in three main sections. So an introductory part, a clustering, and contacts validation. So starting from the introduction, many of you may already know it, but inside our nuclei there is a molecule of DNA, and it is a 2-meter length molecule, which is packed inside the nucleus, which is around more or less 6 micron. We have a lot of different cell types with different cell function in our body, so the way in which the DNA is wrapped inside the nucleus may play an important and crucial role for cell identity and cell function. So let's start from the rules that generates the 3D chromatin organization. There is no simple answer, so not simple rules. It depends upon the scales and at which we are focusing at. But there are two main domains, local features and global features. Regarding local features, we have loops, which are this one. Loops can be structural and functional, usually. Functional loops can be, for example, promoter enhancer, which a contact between promoter enhancer loop can cause initiation of transcription. And then we have structural loops such as cohesin loops that they generate topologically associated domain, which are this kind of structure right here. The role of topologically associated domain, which I refer as TADS, is usually and classically to confine promoter enhancer interaction in order to regulate which promoter will bind which enhancer. And if we move to global features, we have compartment and chromosomal territories. Here we are moving to the most, let's say, 3D part of the chromatin organization. Chromosomal territories are self-explanatory, while compartments are classically two, A compartment and B compartment. A is active chromatin, B is correlates with the repressive one. There are two main approaches to analyze chromatin organization, sequencing approaches and imaging approaches. The most used one and popular one are sequencing approaches because we reached high resolution with also high throughput. And the idea is usually to sequence chromatin regions which are in physical proximity. So the experiment, let's say the most famous one and most important one is IC, but also micro-C. The experiment returned the amount of time two regions of chromatin have been sequenced together, and it returned the contact map of the whole nucleus. And we have a lot of computational tools to analyze this data. Regarding imaging approaches, the idea is to physically identify the region of interest, which are probed. So they are, let's say, suitable for short regions in the chromatin because you have to design the probes. And there are fewer methods, and there are also not many times these computational tools are comparable with the sequencing computational analysis tools. I will focus mainly on the computational tools regarding sequencing-based approaches. There are a lot of issues. The first one is intrinsic within the data. So IC data are really sparse and complex. So identifying which structure is important and also describe the structure is really complex. And the tools tend to focus only either on local or global features. And also they do not usually talk between themselves. So we have got a lot of correlation between the results of tools within and between domains. And we also sometimes lack ground truth in our field. So, okay, this is where I decided to do a PhD on. And this is where I want to place my tool, which is named ICONA, which stands for IC Organization with Network Analysis. We have two main goals. The first one is to provide a unified representation of the chromatin structure. And we do that using generating a nested hierarchical clustering of chromatin through entropy minimization. And the second one is starting from our clustering, so our structure, identify which connections are important and define the structure itself. And we use a method that evaluates the contact uncertainty. Since the second goal and our second approach is based on the first one, we will first focus on clustering, and especially we try to validate the clustering part. So this is the approach. We start with a chromatin network where each node is a chromatin region, and the edge is placed between two nodes and as a weight. The weight represents the number of contacts that have been sequenced by the experiment. And then the assumption is that the best clustering is the one that requires the lower entropy in order to describe the data. So we try with to make a long story short, we select among all the models that we obtain the one with the lowest entropy. We obtain this nested hierarchical clustering that here I'm labeling also the contact map with the same clustering. In order to benchmark the data, I took advantage of this gold standard RC experiment and two cell lines, so IMR90s and GM cells, at 4 different resolutions, which account for the size of the chromatin region that represent a node. The goal is, first of all, to be sure that we can catch local features in the first level of the hierarchy. You can see here we have the contact map, so the raw data, and here hierarchy representing this region. So in order to validate this data, we tried to identify CTCF and RAD21, which is coisin, at the boundaries of TADs, because TADs are enriched at boundaries for this protein. And we found that at the first level of the hierarchy, we have an enrichment for this kind of protein. Between, let's say, 25 and 30% of TAD cluster boundaries at first level are enriched for these proteins. And we are in line with most of the TAD caller. But there is another kind of index that evaluates the quality of cluster, which has been recently proposed, which is this differential presence between 2 histone marks, one activatory that marks actively transcribed region, which is H3K36 trimet, and one is repressive, which is H3K27 trimet. So the present either for one or the other marks a good quality clusters. And using this index, we perform the best among TAD callers. So then we move to global features, especially compartments. And among all the levels of the hierarchy, we identified the same compartment in the cluster. So our cluster were reached either for A or B compartment. But we were not happy with that because the compartments are identified using like computational approaches. So we decided to use also an orthogonal dataset, in particular RepliSeq, which classify chromatin region according to the replication timing, so the time in which they are replicated during the cell cycle. Usually, early replicating regions are associated with A compartments and late replicating regions with B compartment. And here you can see that the regions which replicate in similar stage are found in the same cluster. So at the end, we were at least confident that our results more or less marked the biological signals. So we move to the edge entropy impact score, which score the importance of the contacts for the structure of the chromatin. And we are open for if you have any suggestion for a new name. And we start with the network and the best partition that our model is able to find. And then we shuffle the edges between the clusters. In this way, we can then compute the difference in entropy before and after the edge move. And when the entropy increases, it means that the contact, the edge, is important to define the structure. When the entropy is the same or is lower, it means that the contact is not so important for defining the structure. And in this way, we are able to classify or assign a score, which is entropy impact score, for each contact of our map. There are not so many examples in order to validate this kind of approach because we do not have so many examples of ground structural importance ground truth in IC. But we focused on this one, which is coisin depletion. As I already mentioned, coisin is essential for generating TADs. So coisin depletion is associated to loss of TAD structure, and this has been validated both in imaging and sequencing experiment. And also, but we don't know the rules, coisin depletion is associated with homotypic compartment interaction, much more compartmental strength, let's say. So we applied Icona to this 30 megabase region, and you can see on the top, we have the contact map of wild type and coisin depleted condition. And on the bottom, we can see the edge impact score. And the first thing to notice is that we have this kind of triangular shape TAD structure, which with a high entropy impact score in the wild type condition. While in coenzyme depletion one, we have these long-range interactions which gain importance, which are reminiscent of compartments. But then we zoomed in and we applied Icona to this 3 megabase region right here. Why we selected this region? Because here is the TAD of SOX2, which is really important in mouse embryonic stem cells. In this case, we are using this kind of cell line. This is one of the best described TADs in the literature. And so we applied Icona only on this 3 megabase region. And here we have a shift in entropy importance score. Actually, we lose and we deteriorate the TAD structure in coenzyme depletion condition. And we have this kind of really precise TAD pattern in wild type. In order to characterize a bit better these results, we try to perform this differential analysis. In our field, usually you do like the differences between the two contact maps. So we do the same thing with the entropy impact score. And the first thing that I noticed is that using contacts, you are not able to, let's say, understand which contacts are really enriched in the 30 megabase region compared to the coenzyme depleted one. And here we have a slight increase in the wild type, but really not so strong. But using entropy impact score, we can see that, first of all, the long-range interaction are really increasing and enriched in coenzyme depletion 1. And when we move to the 3 megabase, we have this kind of pattern, which I find really interesting because the intra-TAD contacts are enriched for wild-type cells, while inter-TAD contacts are enriched for coenzyme depleted cells. Then we took advantage of ChIP-seq data, which mark histone marks, in order to classify chromatin regions as active promoter, enhancer, and repressed promoters. And we observed that over the long-range interaction, so 30 megabase region, usually you lose enhancer-enhancer, enhancer-promoter, and promoter-promoter interaction. This is just a log2-fold change between the ratio of those markers. And this, I think this is quite, let's say, expected because you lose the TAD borders, and so promoter enhancers can contact also long-range, with the longer-range interactions. While when we apply Icona only on 3 megabase, here we are focusing on TAD, and this TAD is active in this cell line. And so we increase promoter enhancer interaction in the TAD of SOX2, and we lose some promoter-promoter interaction, which are enriched actually in the coenzyme depleted condition. The final thing that we wanted to do is, since we were lucky enough to have not only the IC data for this kind of region, but also the imaging data, we applied this method also to imaging. And here you have on top IC and imaging on bottom. The data is from the Bottiger lab. And the idea is that, okay, we have this kind of contact map, which is really different from the IC one. But then the edge importance score pattern, the entropy importance score, is really similar between IC and imaging in both conditions, but also in the differential condition, in the differential analysis. So just to conclude, I want to sum up a bit. With Icona, we were able to define a nested hierarchy that is able to capture the multilayered nature of chromatin in the same model, so both local and global features. And with our entropy impact score, we are able to capture the shift between rearrangement upon coenzyme depletion. So why to use this kind of method? Because it's quite, I mean, I think that the first method doing something like that, then if you want to identify TAD, you can use TAD caller, and if you want to identify compartment, you can use compartment callers. If you want to use Icona, you want to identify contacts which drive the 3D organization of the genome, or you want to analyze both sequencing and imaging data with the same kind of approach. The main limitation are the runtimes, because as you may imagine, it is really well written. I didn't write it. It's really effective, but it scales linearly with the number of edges. So if you want to analyze the whole genome, you have to be okay in selecting node with a size of 500 kilobases or 1 megabases. If you want higher resolution, you can go up to 30 megabase region, 40 megabase region. And in conclusion, I want to thank all my lab, my supervisor, Alessio, Davide Cittago and Tiago Peixotto, who helped me so many times for suggestion. And more importantly, I want, because it's here, it's Davide. Davide Bersan helped me with the analysis of ChipSeq data, and also he implemented this spike flow approach, which with spike flow you can like analyze the ChipSeq data using spike-in. It's a novel approach. He is presenting it at process session. His process is the 177. So that's all. I'm ready for the question.
Are there any questions? Please move to the microphones. Is there any questions online? Okay. Go ahead.
Thanks for the great talk. I was just wondering if you looked at looping with your approach.
I didn't look at promoter and answer looping, but I don't know if you are familiar with microcompartments. I looked at microcompartments. Actually, that was a slide about that. I applied, yes, to microcompartments, Icona. Microcompartments are this novel kind of contacts that have been identified by Anders' lab using this region capture micro-C, which is a micro-C over like really short region of the chromatin, and they identified this pattern. And yes, I applied this to region capture micro-C. They state in the article that there are no difference between wild-type transcription inhibition and coenzyme depletion. Actually, we find some shifting in coenzyme depletion importance also for microcompartments contacts. So here is wild-type transcription inhibition and coenzyme depletion. Here we have contact importance. We see that microcompartments are enriched for importance, but in coenzyme depleted condition, we have a lot less important microcompartment contacts. But we didn't try with promoter and answer CTCF right now, because we lack the ground truth. There isn't a clear way to identify which promoter and answer is a real contact right now.
Well, I guess you could compare to existing loop colors and...
Yeah, the problem here is that we cannot we identify all the contacts that drive the shape of the chromatin. So it's difficult because promoter and answer looping caller are really precise on this spot. We call everything that drives the structure. So I don't know.
Thanks.
Thank you. We have another question at one.
Hi, thanks for the great talk. Could you elaborate a little bit more about the imaging data and why you think there is a difference in the maps of contacts, but not in your EIC score?
Sorry, I didn't get the...
Sorry. So can you tell us a bit more about the imaging data? Yeah. And why do you think there is a difference in the patterns you see, but not in your EIC score?
Okay. So the imaging data are... I took it's the ORCA dataset for Bottiger. I think that, and the data is really different here because you can choose to represent the data in different ways. You can use the mean distances between contacts, and here actually I'm using the number of contacts because they provided the threshold at which you can consider a two-region in contact. So I use that kind of threshold. And so just in this way, you know that maybe the difference between contacts stay there because we are generating contacts upon a threshold using imaging. And in sequencing, we are sequencing the close by contacts. But the relationship between contacts and structure is different. So if we are able to identify a structure which is consistent because of the weights and the relationship between connection, and then we base our pipeline upon the structure, which find the best hierarchical model given the entropy minimization. If you believe that, then the contact importance score doesn't, it does not base the results on the weight, but on the structure, which should be similar.
I see. Thank you.
Thank you. Let's thank the speaker again.